So I'm going to draw with you guys how we would solve this problem, and then we're going to use this to kind of draw a map of how we could solve any problem about our membrane potential, our equilibrium potential, and the electrochemical driving force. So we start off with our cell. Okay. And our cell has a membrane potential right now of -70. Okay. So this means that we have a bunch of negative charges inside compared to a bunch of positive charges outside, such that our voltage difference is -70 millivolts would be. Okay, so now what we're trying to figure out is what's happening with potassium. So here we have potassium. We're thinking about potassium inside the cell. Let's switch our colors for a second. Get some blue. Okay, and I've given you in this problem that we know the electrochemical driving force here is pointing outward. So I'm telling you that our electrochemical driving force is telling potassium to move out of the cell. Now we know that the electrochemical driving force is a combination of our chemical driving force and our electrical driving force. So from this information, we can deduce a couple of things. Okay. So the first question, part A, that I asked, is to figure out which direction the electrical driving force on potassium goes. Now for that, we actually don't have to reference this other fact, or for that we do have to reference this other fact. So we know that the inside of this cell has a voltage of -70 millivolts. And we know that potassium is positively charged. So we know that potassium, its electrical gradient, because opposite charges attract, we're going to have an electrical gradient moving into the cell. So we know that the electrical driving force leads that positive charge on potassium to want to join up with that negative charge inside the cell. So our EDF points inward. Now, this is how we manage to get information about our second question, the chemical driving force. So at this point, we know that our sum total of forces points out, and that our electrical driving force points in because opposite charges attract, which means that there's another component of this electrochemical driving force that has to be pointing outward. If the chemical driving force were going the same way as the electrical driving force, then when you added them together, you'd get a force pointing into the cell. So because we can see here that our electrochemical driving force and our electrical driving force are in opposite directions, this tells us that we must have an even greater chemical driving force out of the cell that's opposing that electrical driving force. Okay. And it has to be greater, so that's part C. It has to be stronger, this chemical driving force, because otherwise we wouldn't get movement out. When we added these two forces together, we wouldn't get outer movement unless that chemical driving force is stronger than the electrical driving force. So if they were equal, we'd have no net movement. So we'd have 0 net force. It has to be bigger, and it has to be bigger out. Okay. So this leads us to this final question about the equilibrium potential. So our equilibrium potential is the point at which our electrical driving force and our chemical driving force are equivalent. So in order to get these two forces to be equal, another way that I like to think about this equilibrium point is that basically we're always trying to get this membrane potential to the electrochemical to the equilibrium potential for our ion. So in this case, we can see that our electrochemical driving force is pulling potassium out of the cell. Now, every time potassium moves out of the cell, it's pulling a positive charge from the inside of the cell to the outside of the cell, which means that this number is going to get more and more negative. So if potassium is able to follow its electrochemical driving force, this means that we're going to get a more negative and more negative equilibrium potential. So it's going to travel along with that equilibrium until it hits the equilibrium potential. So in this case, our equilibrium potential is going to be even more negative as it follows this electrochemical driving force until these forces are equivalent. And so the most common equilibrium potential we'll see in our neurons for potassium is -94, I believe. Okay, so this movement, this situation will continue until we get that negative, at which point the force pulling potassium into the cell will be equivalent to our chemical driving force pulling it out of the cell, because as we make the inside of the cell more and more negative, it pulls harder and harder on those charges on the potassium. Questions about this now? We're now going to generalize that to identifying directions of the chemical driving force and the electrical driving force generally. But this is sort of how you would work it out, what we're about to go over.
So what number is 94 again? That's like where the equilibrium can start?
That's the equilibrium potential for potassium. So that tells you that's the point at which the chemical driving force and the electrical driving force are equal, which means we'd have no movement of our ion.
Is it different for like different things like other than potassium?
Yeah, absolutely. So when we're paying attention to our equilibrium potential, it's going to be for a specific ion, basically, because it factors in this chemical driving force, which is dependent on the gradient of that specific ion. Remember that the chemical driving force is specific, like to each ion or to each molecule, whereas the electrical driving force is generalized for just positive or negative. So yes.
Okay, sounds good.
Mm-hmm. Sodium happens to have a positive one. We'll see that one as well.
Okay. Are those two numbers, like -70 and -94, like, are they supposed to like add up to a specific number, or are they just kind of like specific to potassium in this case, or the specific example?
They're specific to this example and specific to potassium. So we're not going to need to add these together or subtract them, but we kind of want to compare which one's bigger and which one's smaller, because that tells us something about the relationship of these forces, if that makes sense. Because when we're looking at the equilibrium potential for an ion, this -94 number, that's our equilibrium potential. So that's telling us that potassium is going to want to move until we get to -94 millivolts. So that's why we would want to reference this other number, the membrane potential, because if we're not at -94, we know potassium has some pull on it in some direction. And by looking at what that value is exactly, we can tell which direction that pull is.
So then in that case, you would say that the equilibrium potential for potassium is stronger, right? Than the membrane potential? Okay.
Yep. Okay. So we're going to draw out a generalized chart comparing the chemical driving force, the electrical driving force, and thinking about these equilibrium potentials and membrane potentials, because if you work out this type of example for all our scenarios, you kind of get a flow chart that, I'm not telling you to memorize it, but you could make a flow chart working through examples like this and figure out the relationship between all these numbers. And it'll be the same for all sorts of different examples, right? If our cell is at -60 versus our cell is at -100, there are relationships that follow from that. But I wanted to go through an example first because you don't actually need to memorize anything if you understand the principles behind it, if you understand how these things relate. Okay, so I think I can actually type. Is that right? Okay, so we're looking at determining, yeah, way better, the direction of the ECDFs, or electrochemical driving force. And the easiest way to do this is if we happen to know what our CDF and EDF are, so what our chemical driving force and electrical driving force are. And if we don't know it, we should always read the problem to see if we can figure it out. So first we're going to want to identify those directions. So if we can identify those directions, we're going to get to our first split. Okay. So our first situation, there we go. Okay. So our first scenario is going to be if they're the same direction. So if they're the same direction, so say the CDF and the EDF are both pointing into the cell, then we don't have a problem determining the direction of the electrochemical driving force, because since they're both pointing the same direction, our electrochemical driving force is going to be in that direction. But if they're in opposite directions, we're going to have to do a little more thinking. We're going to have to find some other numbers to tell us more facts. So they're in opposite directions. And one of the things we can do if they're in opposite directions is that we can compare that membrane potential with the equilibrium potential. So at this point, we can compare the sizes. So we abbreviate that EX, and the X is really what subs in for potassium or sodium or whatever. So we'd have like ENA or EK or something like that, depending on specifically what we're tracking. So we're comparing the sizes of that equilibrium potential and the membrane potential. So now we're going to have three options. The simplest one is what I'm going to start with. So in our simplest case, they're equivalent, right? So our simplest option is if the equilibrium potential and the membrane potential are the same. So say they're both -94, working with that example from before. Our electrochemical driving force is equal to 0. So our chemical driving force exactly opposes the electrical driving force. That's our equilibrium potential, which means that we have no electrochemical driving force because these forces are directly in opposition. So this also means our ions are at equilibrium. So there's no net movement. But a lot of the time we're at a different membrane potential than our equilibrium potential. And sometimes we'll be holding our cells so that these equilibrium potentials and membrane potentials are different, so that we can allow all our ions in a flood or let them out in a flood. So it's not always a problem if they're different, but we do always want to know which way our forces are going to point, which way those ions want to go. So here we're going to start out our scenario just now where the equilibrium potential is stronger than the membrane potential. So like in the example we just went through, our equilibrium potential is stronger than the membrane potential. This actually means that our chemical driving force is stronger than our electrical driving force. Because our chemical driving force is stronger than our electrical driving force, this means that our electrochemical driving force will follow the chemical driving force. Now that seems like a random series of facts, but we just worked out why that is true. So we just went through an example of an electrochemical driving force following a chemical driving force, resulting in an equilibrium potential that's stronger than the membrane potential. So that's why we did that drawing first. So we also will have the reverse sometimes. So sometimes So once we've identified the electrochemical driving force and its direction, really the reason we were doing that for our purposes for right now was just to figure out whether something's going to be passively transported or actively transported. So that was a very long way to talk about some complicated facts about gradients, but we have to do all that in order to figure out which direction ions are going to move. So when we're talking about ions, we need to think about the electrochemical driving force that we just went through. But a separate thing to note is that we will see the movement of lots of things that aren't charged, right? So like oxygen, things like glucose don't have a charge. So in those cases, our life is a little simpler because we only have to think about a chemical driving force. So when there is a charge. So we had to go through all that to understand ions, but when by the three general factors that influence the rate at which transport is going to happen across a membrane. And we're going to identify the factors that affect the permeability of membranes. So when we say rate of transport, what we've just been talking about is the direction things are going to go. What we now are thinking about is how fast are they going to get there. So when we talk about this, we often talk about something called flux. So flux is really just movement. And so whenever we have molecules floating around inside the cell, floating around outside the cell, we're going to have a little movement across the membrane and a little movement back across the membrane if the membrane is permeable to that substance. When we're at diffusional equilibrium, the amount of that molecule moving out of the cell is the same as the amount of that molecule moving into the cell. So we say that there's no net transfer of molecules. So we simplify our lives. We can have a little bit moving in and out of the cell back and forth, but they cancel each other out. So we just basically think of it as no movement. Okay. So that's our diffusional equilibrium. And that's what molecules try to move towards, right? We try to move so that we're at diffusional equilibrium, which would look like equivalent concentrations on either side of the membrane. If we have unequal concentrations on either side of the membrane, so we were just talking about the chemical driving force, this means we'll have a chemical driving force if we have a difference in concentration, which means that the movement of molecules in one direction, so down the gradient, it's going to be more than the movement of molecules against the gradient. Okay. And the reason there's any against the gradient is just by random chance, basically. Movement of molecules is kind of probabilistic rather than always individually tightly controlled, assuming always that the membrane is permeable to that molecule. Okay, so when we have a difference in concentration, we're going to have more movement down the gradient than up the gradient. So our movement from high to low is going to be greater than low to high. So we call that net flux. So the net flux, or the net movement of molecules, will be towards the lower concentration. So that's our downhill movement down a gradient. This is how diffusion works. Diffusion is a form of passive transport. So it's passive because we don't have to put in any energy. Okay. So passive transport has some specific characteristics. So we just covered that it's downhill movement, right? So down a concentration gradient from an area of high to low concentration. That's our net flux, is downhill. Requires no energy because it's obeying the laws of thermodynamics. We don't need energy for something to spread out and get more disordered. So moving down this gradient is energetically favorable, which means that this is also spontaneous. So this is just how the molecules move. Spontaneous, we didn't need to do anything to make them do this. We classify passive transport as basically one of three types. And the reason we do this is not that they have different directions, but it's basically referring to how they get across the membrane. So we have three ways we can get across a membrane depending on what molecule we're talking about. So those three types are simple diffusion, facilitated diffusion, and diffusion through channels. And we're going to go through each of those. Okay. So starting with simple diffusion, simple diffusion happens directly across the membrane bilayer. So we're talking about something that can get through those phospholipids, something that can get through that strong non-polar region in the center of the bilayer. So we don't need any membrane proteins. These molecules can go directly across. If something goes via simple diffusion, we don't have any transporters, we don't have any channels. We're just moving directly between phospholipids. So things that undergo simple diffusion would be things like oxygen, that just diffuse directly across the membrane. So they're going to be small, they're going to be nonpolar, they're not going to be charged, they diffuse directly across the membrane. There are still factors that affect how fast they go. It's not all instantaneous. The first is going to be the magnitude of the driving force. So because they're going directly through that lipid bilayer, they're probably not ions, they're probably not charged. So we're thinking here the chemical driving force. So how big that driving force is going to make a difference for how fast. If we have a bigger driving force, we're going to go faster. The surface area of the membrane also makes a difference, because simple diffusion basically relies on those molecules bumping into the membrane and pushing through. They don't have to push that hard because the membrane is permeable to these molecules, but they do have to come into contact with the membrane. So if we have more membrane, they're more likely to contact the membrane, which means they're more likely to get in, which means that we'll have a higher rate of diffusion across that membrane if there's a greater surface area of that membrane. Final thing is membrane permeability. Some molecules have an easier time getting across the lipid bilayer. So just because they can get across doesn't mean they all get across equally easily. So if the membrane is more permeable to that molecule, our rate's going to go up. Okay. So we see this nice straight line. Love to see it, right? So here's our magnitude of the driving force and its effect on our rate, which here we're expressing in terms of net flux, so that net movement of molecules. As our concentration gradient goes up, right? So we have a bigger difference in concentration inside versus outside the cell, bigger difference, more molecules moving. This is a picture of some cell membranes. So this illustrates our membrane surface area. Different cells are going to have more membrane surface area. So those little pink lines are some membranes. And in particular, when we look at lungs or capillaries, we're going to really be specialized for allowing diffusion to occur across those membranes. So in the lungs and in the capillaries, honestly, we want a lot of oxygen to cross those membranes. So we're going to have a high surface area in those areas so that we can move those molecules rapidly across the bilayer. So higher membrane surface area, more diffusion. So our final bit is the permeability here. These should really be tabbed out. So membrane permeability factors, there are a couple of them that affect the rate. So the simple part, right, is if our membrane is more permeable, we're going to have more flux. So if this membrane is more permeable, it's easier for our molecule to get across the membrane, which means our rate is higher. But those membrane permeability factors, the membrane permeability is dependent on often the lipid solubility of the substance that's diffusing. So our phospholipid bilayer, right, there's the word lipid in there. If something is highly lipid soluble, that means that it's able to get along really well with lipids, which means it's more able to cross the membrane. It's going to be easier for it. And the second piece of this is the size and shape of the diffusing particles. So smaller things in simpler shapes have an easier time fitting between those phospholipids, basically. So they are able to get through the membrane more easily, which means that the membrane will be more permeable to those things. So that's all this is saying, is that something that's lipid soluble and probably small and simple shaped has an easier time getting across the membrane, which means our rate of diffusion will be higher. Another factor that does theoretically affect the rate of diffusion is temperature. But temperature is a regulated variable in humans, you know, and in living organisms. So this isn't really relevant in human physiology. Our temperature is kept in a really tight range, so we shouldn't have too much temperature variability affecting rate of diffusion in human bodies. What we will see differences in is the thickness of a membrane. And that's basically just to say that if we have a thin little membrane, it's easier to get across. We don't have as far to go. We don't have to cross that big of a barrier. If we have a thicker membrane, it's harder to get across. So again, we'll see that places like capillaries have thin little membranes because we're trying to up that rate, right? So if we're trying to get stuff to diffuse really quickly in some part of the body, we're going to see a thin membrane, high surface area. We're going to try to let things across easily, so long as they're the right things. Okay, so these are all factors for simple diffusion. So just simple diffusion directly across the membrane, no proteins. You can practice with these later. These checkpoint questions should be fairly straightforward. We're going to move on to thinking about carriers and channels, our other two types of passive diffusion that we will see. So passive transport through carrier is called facilitated diffusion. Now, a carrier is an integral membrane protein. When we talked about membranes, we talked about the fact that we have some proteins that are embedded in that membrane. Some of them go all the way through. So we have an end on the inside of the cell, we have an area going through the non-polar region, and then we have an area on the outside of the cell. So that's going to be true about all our integral proteins. Our carriers specifically basically have little pockets that flip or change shape. So our carriers are transmembrane proteins. So the reason they're allowed to facilitate or make easier diffusion is that they do have a connection to both the outside and inside. Wouldn't help us very much if they only touched one side. They're going to have binding sites for specific particles. Guys, I see my microphone suddenly just changed. Can you still hear me? Okay, good. Okay, so our carriers are going to be specific to specific molecules. So in this example, you can see this is labeled a glucose carrier protein. That means it's going to let glucose through. It's not going to let sodium through, for example. So when we're looking at carrier proteins, they're going to be specific to a specific substance. And the way they work is that that substance, that particle, whatever it is, binds in to one side of our carrier. Then the binding site changes shape. So we talked about how proteins can change shape when we talked about protein structure and when we talked about enzymes. This is an analogous kind of situation. So our binding site changes shape, which flips what side of the protein is open. So we had glucose here bind into the pocket in the extracellular fluid in this carrier protein. Then we can see once the glucose has bound, this just changed the shape of that protein. And so now this little pocket area is on the inside of the cell, which means glucose passes across. It's now free to intermingle with our intracellular fluid. So just be careful. I know that changing shape kind of seems like it would have to involve energy, but it doesn't in the case of facilitated diffusion. This is just something that happens when these two things come into contact, when the particle comes into contact with this protein. So when we're talking facilitated diffusion, we do have this shape change, we do have this orientation change, but we don't need energy for that to occur. This is still passive transport. Binding is going to occur on one side at a time in facilitated diffusion because of the way this looks, because of this sort of pocketness, right? The pocket's only facing one side of the cell at a time. Okay. So when we're looking at rates of facilitated diffusion, one thing that is true about these carrier proteins is that they have rates of transport, basically, right? It takes some time for them to flip. It takes some time for them to go back to their original orientation. So there will be a speed at which those carrier proteins work. So that's going to affect the rate of transport for facilitated diffusion. Another thing that's going to affect our rate of facilitated diffusion is the number of carriers in the membrane. So this is kind of like with simple diffusion, where we're talking about how surface area affects the rate of simple diffusion. So our analogy here in facilitated diffusion is the number of carriers, because we're still relying on those particles coming into contact with the membrane. But in this case, specifically, they have to come into contact with one of their carriers on the membrane. So if you have more carriers, our rate is going to go up, right? Because these particles are more likely to contact those carriers. And if you have fewer carriers, you don't have that many of that protein, of that glucose transporter, for example, then our rate is going to go down because they have to find a carrier. They have to come into contact with it in order to get across our cell membrane. And finally, our concentration gradient also has an effect on our rate. And that's because, right, imagine that we only have a little glucose outside the cell. It's trying to get in, has to bump into a carrier to do it. It's going to have to float around for a while before it contacts the carrier. But then imagine a different situation where we have a ton of glucose surrounding that cell. The probability that that glucose is going to contact a carrier is higher because there's just more of it. It's all bouncing around, it's bumping around. So the concentration gradient is going to have an effect on our rate, just like it had an effect on our rate of simple diffusion. So here we see an image of what this looks like. So as our concentration gradient is going up, we see an increase in the rate. But the difference here from our simple diffusion is that we see this sort of level off, right? So before we had that theoretical kind of just straight line across the center here. But what we're seeing here is that as we have a bigger and bigger concentration gradient, our rate is going up until all those carriers are full. So once we are using all the carriers on the membrane, we can't increase that rate anymore because they're all already working. We have to wait for one to have that pocket empty and facing the outside of the cell. So carriers can get saturated at a certain point, and that's why we see this flattening out point. If we increase the number of carriers, we'd sort of raise this curve and we'd have a flattening out somewhere else. I'll stop after this, but this example, I've been using glucose as an example. So an actual example in your textbook and in real life is GLUT4, which is a glucose transporter. So GLUT4 molecules are made in the rough endoplasmic reticulum. They're packaged into vesicles and they're inserted in the membrane. That's how you end up with this transporter in the membrane. So we can vary that process basically. So we can have our rough ER make more GLUT4, or we can have it make less GLUT4. So we can change the number of that carrier protein in our membrane. Specifically, insulin stimulates the insertion of GLUT4 into the membrane. So when we're in the presence of insulin, we're going to be making more GLUT4. We're going to be ending up with more of this carrier in our membrane. Now, this works a little differently in diabetes. So in diabetes, specifically diabetes mellitus, where insulin isn't working, when we have a lack of insulin, this means we don't have that transporter inserted into the membrane. So we stay with a sort of low baseline level of this glucose transporter, which means that we can't get glucose into our cells, really, because that transporter, that carrier protein, it's full. We only had a couple, so no matter how much glucose we have, it's going to get saturated. And when we have this lower level of glucose uptake, it means that that glucose just stays in the blood. So your blood sugar is going to rise in this case because we can't pull that glucose out of the blood and into the cells. Okay. So that is a clinical example of why this matters, basically, why we care about the rate of transport and how we can vary it. So we'll pick up here on Wednesday. As usual, I'm going to stick around, but you guys are free to go.